Hello and welcome to this lesson for Advanced Higher Biology, which in this one is going to be looking at the cell cycle. So for this one, you need to be able to understand the need for the cell cycle and the phases that make it up. You need to be able to name the two phases of the cycle, list the subphases involved in interphase, list and describe what happens during both stages of the mitotic phase, and list the overall stages of mitosis and describe what happens in each of them. So to begin with, a little recap on microtubules and the fact that microtubules are found in all eukaryotic cells, and they originate from the microtubule organizing center, or MTOC, or centrosome. So this centrosome is found near the nucleus and contains centrioles, which are the site of microtubule synthesis within the centrosome. Microtubules are quite important when it comes to cell division and the cell cycle, as they are what forms the spindle fibres. They're also responsible for helping the cell remodel the cell's cytoskeleton, which we're going to need to be able to do if we're going to form two new daughter cells. So the spindle fibres are active during cell division. Shown here, we can see them in green, and the spindle fibres are helping to pull the chromosomes, which are labelled blue here, to their varying points across the cell. They're being pulled to the poles in the cell by these spindle fibres here, which are made up of microtubules and have come from those centrioles being produced within the centrosome. Now, during cell growth and division, we require that in order that all organisms can come to be as a result of that repeated sequence of cell growth and division. The overall sequence of events a cell goes through allow it to duplicate its contents and divide, and that's known as the cell cycle. The products of the cell cycle are two genetically identical daughter cells. In single-celled prokaryotes, this would be the process by which those organisms would reproduce. However, in eukaryotes, this is going to allow for growth and repair by replacing worn-out cells. So some cells will need replacing quicker than others. For example, the absorptive cells in the small intestine are replaced every 5 to 6 days, while on the other hand, you've got red blood cells being replaced every 120 days or so. And that's going to vary depending on the role of each individual cell. Now, the cell cycle consists of two main phases. We have interphase, which is a period of growth, and we have the mitotic phase, which is the time at which the cell divides. And as you can see, interphase lasts much longer than the mitotic phase does. If you were to look at a group of cells by microscopy, you'll only see a small proportion of them in the mitotic phase. Most of them will kind of look like they're doing nothing. However, although you can't see anything taking place, describing interphase as the resting phase isn't quite true. Interphase is an active period of growth, so there's a lot actually happening during interphase. We have, for example, protein synthesis, organelles are being produced, and we're also replicating chromosomes. So as a result, interphase itself has been divided into three subphases. The first one is the first growth phase, which is also known as G1. And that's our initial growth period where we are making proteins and organelles for the cells to use. Our next phase is our S phase, and that's where DNA is being replicated in preparation for mitosis to occur. In our final phase, which is a second growth phase referred to as G2, there is another growth period and we've got proteins and organelles being synthesized again. At the end of G2, our cells are going to enter the mitotic phase, which is split into the stages of mitosis and also a second final stage called cytokinesis. And we need to look at the stages of mitosis in detail just now to have a little look at what's happening in each one of those individual phases. So to begin with, we're going to start off with looking at prophase, which is our initial section of mitosis. When we have prophase, we have our DNA condensing into chromosomes, and each chromosome will now consist of two sister chromatids. The nuclear membrane surrounding the chromosomes is going to start to break down, We have spindle microtubules extending from the mTOC by polymerization and attaching to chromosomes via their kinetochores, which is these sections here. This is going to occur in the centromere region before we head into metaphase. So during metaphase, we have our chromosomes aligned across the metaphase plate. So this was previously referred to as the equator when you looked at it in National 5. So the arrangement of chromosomes across this plate is referred to as the metaphase plate. And that's just getting the chromosomes essentially to get themselves lined up and organised so that they're pointing in the right directions. We've got one sister chromatid facing that way and another one going that way. We then head into anaphase, and again we've got our spindle microfibers here which are attached onto these centrioles, and what they're doing is they're being depolymerized. Remember, they are made up of microtubules, so our spindle fibers being depolymerized. We're losing those tubulin dimers and they're being pulled apart as a result. So our chromosomes are now being pulled apart. So we've got one sister chromatid heading in this direction and the other one going in this direction. So they're pulled to opposite poles. And that leads us into telophase. And during telophase, our chromosomes are going to start decondensing and the nuclear membrane is going to form around them. This finishes off with the second stage of mitosis, which is cytokinesis, and that's where we have the separation of the cytoplasm, the cytoskeleton, into two daughter cells. And it's important to note just that microtubules are quite important throughout this process. The spindle is beginning to form at prophase and is organised by the centrosomes at the two poles of the cell. Spindle fibres then allow the alignment of the chromosomes at the metaphase plate, but they also help separate the chromatids to the opposite poles. And after that, we then have spindle fibres playing an important role of the daughter nuclei. So our new nuclei that are being formed in this section are being helped to form by those spindle fibres, and that allows our separated chromosomes to group at opposite ends of the cell and allows our new nuclear membranes to form, producing our new cells. So from that, you should know that we have two different phases of the cell cycle. We have interphase and we have the mitotic phase. That in the phases involved in interphase, we have our G1 growth phase, our S phase, and our G2 growth phase. In the stages of the mitotic phase, we have mitosis and we have cytokinesis. And that during mitosis itself, we have prophase, metaphase, anaphase, and telophase. And during prophase, we have our initial condensing of the chromosomes and formation of the microtubules. In metaphase, we have the chromosomes aligning at the metaphase plate. During anaphase, our spindle microtubules are being shortened by depolymerization and the chromosomes being pulled to opposite pores. And during telophase, we have the decondensing of the chromosomes and nuclear membranes forming around them. Finally, we have cytokinesis, which is where our cytoplasm and cytoskeleton are separated, forming two daughter cells. As ever, feel free to ask any questions on this at all as we go over it in class.